So now I'm going to talk about the parts inside of a eukaryotic cell and the overall structure of a eukaryotic cell. So the first thing I need to mention is the outer boundary of a cell is called the cell membrane. And in this picture, that's this pastel or light colored covering on this half cell drawing here that an artist put together. Um, the material that's inside of the cell membrane includes this tan material, which is called the cytoplasm. That's over here, cytoplasm, also called protoplasm. And it's jelly-like material. It has enzymes in it. It's mostly water. It has vitamins, minerals like sodium and lithium and chlorine and whatnot. It also has proteins, carbohydrates, lipids, and not much nucleic acid. Most of the nucleic acid's in here in the nucleus, but there's some RNA out here running around. And then all these other colored objects that are floating around in the cytoplasm, those are the organelles. And so there's all kinds of activity going on out here, but the boundary of life for a cell is the cell membrane. Outside of the cell is not alive. The cell, made up by its cell membrane, everything inside, is alive. And I should mention that the size of a cell is limited by its surface area to volume ratio. And as it turns out, for very small cells, the surface area of this tan material out here can actually keep the cytoplasm supplied with nutrients, oxygen, get rid of carbon dioxide, etc. But as a cell gets bigger, it turns out the volume gets bigger faster than the surface does. And so that's why there really are no big cells on Earth. There are no cells the size of a chicken egg because cells that big cannot survive. The movement of materials through the membrane for something the size of a chicken egg would be insufficient for the amount of activity going on inside the cytoplasm of that giant cell. So there are no giant cells like that on Earth. Cells are tiny because of the surface area to volume restrictions. Now, the holy grail of all organelles is the nucleus. And so these are three cells here. These are actually human nerve cells, if I'm not mistaken. This is a typical nucleus. This is a typical nucleus. This is not. This is an atypical or a non-typical nucleus. These two nuclei have DNA in them, and it's in chromatin form. Chromatin is DNA that's not tightly packed, and so it looks like this. It looks just multicolored, light and dark, and looks like just things are scattered. Now, these, on the other hand, are chromosomes. Chromosomes are only found in dividing cells, and this cell here was preparing to divide. So this brightly stained stuff here and the brightly stained stuff there, those are tightly packed DNA molecules. And that is only present for a cell in cell division. So this particular cell has DNA, and these other two have DNA, but these cells have DNA in the chromatin form, and this cell has DNA in the chromosome form. Now, the nucleus is notorious for producing RNA, and RNA is what tells the rest of the cell what to do, and I will get more into the details of that in a minute. And then there's a membrane. You can't see the membrane well around this because it's dissolving, and that's what happens in a dividing cell. These cells have a definite nuclear membrane. The nuclear membrane's got big holes in it to allow RNA in and out and to allow ribosomes in and out because the nucleus produces ribosomes. Actually, the nucleolus, which is a small region inside the nucleus, actually produces the ribosomes. So that is the story of the nucleus. Okay, it's the holy grail of a cell. It is where the DNA is, which is really controlling all of the cell's activity. The next organelle is the ribosome. Now, the ribosome is really the kind of the smallest organelle we're going to talk about. Ribosomes are the site of protein synthesis. It's where you make your proteins. Now, it turns out that your DNA is actually telling your ribosomes what proteins to make via RNA. So the DNA sends out RNA to the ribosomes. These are the ribosomes. And proportionately, these ribosomes are much smaller than these nuclei. If this was drawn proportionately, the ribosomes are not even the size of that little dot at the tip of my mouse up here in this picture. Ribosomes are absolutely tiny, tiny organelles. They're the tiniest organelle in our discussion here, and they're really only visible just barely with the transmission electron microscope, and even then they're fuzzy. And so ribosomes are made of RNA and protein, and they're made of two pieces, a big piece and a small piece. They call them subunits. So there's like a big subunit and a small subunit, or a large subunit and a small subunit. There is no membrane around a ribosome. And what they do is they attach to RNA, and that's what this black thing here is going through the ribosome. This is the big ribosome subunit. The yellow piece is the small ribosome subunit. And these pieces that are bouncing in here are bringing amino acids. That's a polypeptide, that little gray strip that's going up in the air. That's a polypeptide that the ribosome is producing. And the polypeptide right now is going to be shoved. It was shoved into the ER, but they're not showing that at this moment in the animation. So ribosomes are your protein factories, and every cell's got hundreds of them, because every cell is busy making proteins all the time. Now, the next organelles are the ER, or the endoplasmic reticulum, and the Golgi apparatus. And these two kind of work together. The ER is this orange membrane stuff here, and it's these wavy lines here. Basically, if you took a big old garbage bag and you wadded it all up into a really tight wad in your hand, crumpling it, you'd have yourself a good analogy for endoplasmic reticulum. ER is connected to the nucleus, connected to those big pores. So things that go in and out of the nucleus, like RNA, often go in and out of the nucleus through the ER. So the ER kind of helps transport things in and out of the nucleus. So the ER encloses a continuous space, just like a bag. It doesn't have holes in it. It's actually enclosing a space. The ER's job, well, actually, there's two kinds of ER. There's smooth ER and rough ER. Smooth ER detoxifies substances, and your liver's got a lot of it there. Anytime you eat or drink something that may not be good for you, your liver's job is to make it less toxic. Alcohol, for example, is detoxified by your liver on smooth ER. But in addition, there's another kind of ER called rough ER. And rough ER has ribosomes on it. And that's what these little blue dots are on this orange ER. Ribosomes that are stuck on ER shove the proteins they make into the ER, so that then those proteins can go to the Golgi apparatus. Now, the Golgi apparatus's job is to modify proteins that are being sent to it. Turns out proteins are really complicated, and the ribosome can put them together. In other words, it can string amino acids together, but it cannot finish their three-dimensional shape. That's what the Golgi's for. So the Golgi, it's sort of like pasting little pieces of paper together to make strings, and then you pass that string to somebody, and then somebody takes the string and folds it in and creates knots out of it, and then makes a nice looking shape out of it. It's sort of like taking a long tubular balloon and then watching somebody bend it and fold it and turning it into something that looks like a dog. That's what the Golgi does to the polypeptides manufactured by the ribosomes. So the Golgi apparatus, unlike the ER, is not one continuous bag. Instead, it's multiple bags. It's like Ziploc bags stacked on top of each other. And each bag tends to do different things to the proteins that are coming in. So the little bags will send proteins to the bigger bags, which send proteins to the bigger bags. And a lot of the proteins that come out of the Golgi apparatus will then be sent to the cell membrane, and they're exported out. Insulin, for example, that gets into the human bloodstream is made this way. Insulin is manufactured on rough ER. The rough ER passes the insulin protein, which doesn't work yet, to the Golgi apparatus. The Golgi apparatus actually puts together two polypeptides to make insulin, and then folds them up to make them work. And then it sends them out of your pancreas cells by sending the little packages of insulin proteins to the cell membrane, and they invert. They pop inside out, and the insulin's dumped into your bloodstream. So there's an animation down below that kind of demonstrates this. This is a TEM. This is a nucleus here, and this is ER up here. Down here, this is a Golgi apparatus right here. And these are vesicles of protein that the Golgi is sending out. So what we have in this little animation here is the nucleus is manufacturing ribosomes, and you can see them come out of the nucleus and they go on the rough ER. The rough ER then is making proteins, and the proteins are being packaged in these little vesicles, and the little vesicles are traveling right there. They travel to the Golgi, and then they go from sac to sac. And the Golgi's job then is to modify those proteins and it does. And then it sends the proteins to the cell membrane. You'll notice right here at the tip of the mouse, the proteins pop outward and they end up outside the cell. This is how insulin is exported from your pancreas cells into your bloodstream. And as a matter of fact, most of your protein hormones that get into your blood are sent into your bloodstream this way. Now, the next organelle to talk about are the lysosomes. Not much to say about lysosomes. They're an essential organelle. They're bags. They look like these. Some of these could be lysosomes, okay? These little bags that you see on this transmission electron micrograph. All lysosomes do is break down worn out organelles. That's their job. So any old organelles that are destined to be broken down, they bind with a lysosome. The lysosome then dissolves them. Lysosomes are also critically important for white blood cells to destroy microbes. So whenever you get sick and you have bacteria growing in your lungs, say you get a pneumonia, your white blood cells eat the bacteria, but then the bacteria have to be destroyed. Otherwise, they'll grow inside the white blood cells. Your white blood cells use your lysosomes to literally almost like bleach. They don't use bleach, they use oxygen. It's like OxyClean. And they sort of destroy the bacteria by oxidizing them with oxygen. The next organelles to talk about are vacuoles. In animal cells, vacuoles look like these little bubbles. And it's very hard to tell a vacuole from a lysosome, except that the vacuoles are usually brighter colored like this. Lysosomes are usually darker like that, and vacuoles are bigger, lysosomes are smaller. Plant cells, though, have often a big vacuole, and that's what this big blue thing here is on the inside here of this plant cell. And basically they store water, and then they'll store also some salts, proteins, and carbohydrates, but mostly water. Some plants actually store poisons in their vacuoles so that if an animal eats the plant part, the animal will die. So some plants are famous for that, like hemlock is a famous toxic plant. Most plants, like hemlock, put their poison inside of their vacuoles to sort of make an animal pay should the animal eat on them. So next ones, the most important maybe organelles on Earth, mitochondria and chloroplasts. Lynn Margulis first proposed, as I said before, that these two organelles were probably at one time free-living microbes, and then they began to live inside of other microbes, and that was the origin of eukaryotes. Before that, everything was a prokaryote. These little green balls that you see in this picture, those are chloroplasts. And these big round objects are mitochondria. So there are two mitochondria in this picture on the right, but there must be several hundred chloroplasts in this picture on the left. Chloroplasts do the photosynthesis on Earth. They absorb sunlight and they convert it to chemical energy. And as they do that, they take carbon dioxide and water to store the energy, and they reformulate the carbon dioxide and water into sugar and oxygen. And the sugar actually stores the energy of the sunlight. So every time you eat anything, you're eating stored sunlight, manufactured or converted, I should say, by chloroplasts on some plant somewhere. Mitochondria, on the other hand, are the engines that you and plants use to take the stored energy of carbohydrates from chloroplasts and they turn it into usable organelle fuel. Organelles can't run on carbohydrate fuel. They run on what is called ATP fuel. And that'll be something we'll talk about in the next chapter. But mitochondria take carbohydrates and turn it into ATP fuel for energy. It's, it's like your car, your automobiles that you drive to school, most of them run on gasoline. And gasoline is not just found anywhere. In fact, it's really not found anywhere on Earth. We have to make it from crude oil. So we pump crude out of the ground and we transform it into gasoline so that the car will run on it. Well, likewise, your car is like an organelle in your cell. It won't run on just anything. It needs gasoline. And the gas station is a nice dispensing station. You can think of mitochondria as sort of gasoline or energy processing centers that take in raw energy that's really not usable by anything and turning it into usable fuel for all the other organelles in the cell. I should mention, mitochondria actually have DNA in them, and so do chloroplasts. And you can actually do genetic lineage studies on mitochondrial DNA. And oddly, mitochondria are only inherited from mothers, not fathers. And so it's easier to trace mitochondrial DNA through family lines than it is than nuclear DNA. But we'll talk about that at a later time. Some of the last organelles here that are cell structures, the cytoskeleton is stained here. That's these green and red fibers that you see scattered in the cell. Actin filaments are in red, microtubules are in green, and the nuclei are blue. So you'll notice the nucleus here has none of these filaments in it. But the microtubules, which are literally small tubes like pipes, are in green, and the red stuff are more like rope. Now, the cytoskeleton is basically just about all protein, and it's what holds a cell together and gives it shape, because cells are super delicate. So if you pull on them and then they didn't have any, if you didn't have any kind of reinforcing protein in your muscle cells, when you go to pick something up, your muscle will just snap in half. But when you lift something, you're pulling on proteins inside of each cell. So the proteins inside of each cell are cemented also to proteins in neighboring cells. So your muscle is actually a giant interconnected network of actin and myosin protein fibers running throughout the cell that give the muscle its strength. So microtubules are hollow proteins. Microtubules, you'll see in the next animation, are important in cilia and flagella, and they provide infrastructure inside of a cell. Microfilaments are solid threads of protein, and they give lots of cells their shape. So cytoskeletons give cells solidity, structure, and strength. And then the last topic here before we go on to membranes is about cilia and flagella. Cilia and flagella are made up of microtubules, not filaments, so tubes, and they protrude through a cell membrane, and they are used for wiggling. Sperm are pushed by flagella, but many microbes pull with their flagella. This is a euglena pulling itself along with a flagella. The human trachea, that's your windpipe, and your bronchi, which is the branching tubes from your windpipe, are coated by cells with flagella that push mucus up out of your lungs and your windpipe so that it doesn't accumulate down there. Smoking kills those cells, and that's why people who smoke get black deposits in the bottoms of their lungs that can't come out. It just sits there, and then that's what ends up causing cancer in later years. So cilia are little short hairs that are found on some microbes that are used like little oars on a boat to propel cells through the water. So cilia and flagella are good for moving cells through water. And most human cells don't have them, but just a few do. And now in the next lecture, we'll move on to boundaries of the cell.